Okay, recording. So let's start. So welcome to the November 30th, 2022 Joint Food Ontology Discussion Group. And today we've got Tarini Naravane talking about a recent paper that she did and where she's going with that work. So over to you, Tarini.
Thanks. Thanks for being here, making time for this today. And please, I look forward to hearing your comments, any feedback, any suggestions, how you think this work might fit into the various things that you are doing. So any discussion after this is very welcome. I'm at UC Davis in the Biological Systems Engineering Department. And I am working to graduate before the spring summer of next year. So as the title read, this work is about creating a machine learning model to predict the nutritional profile after food processing. And the challenges here is that foods are very complex materials. They have composition that we don't know about completely, and we almost know nothing about the structure. And both the composition and the structure plays a role in what happens when you cook food, what the nutrient loss is like, how the chemical forms of the nutrients or the compounds, how they change, and then, of course, all the various sensory effects, the texture, the flavor and stuff, all that is very hard to predict. And the reason is that it's a multi-parametric problem of unknown dimensions. So the fact is that we don't really know how many factors we need to consider. I mean, of course, this hasn't stopped us from processing food, from eating food, from being able to provide nutrient labels. And so I'll get into how we do that now and what machine learning offers. The other challenge is that experimental determination of whatever the effect is costly, time-consuming, reductionist, meaning that it's limited in some experimental scope. And it's not a scalable approach. We can't measure everything forever, like everything about everything. So the current computational solutions have to be simplified models. As I said, we don't know what the dimensions are, and experimental determination has to be reductionist. So the simplified, there's two models, and one of them will be the baseline model that I'm comparing the prediction against. So the first is a kinetic model where the concentration of any nutrient is a function of the time and the temperature that you're cooking it for. This is not scalable, and this is very specific to certain compounds and in certain foods. The retention factor method is much more general. For example, retention of 50% of some nutrient in some category of vegetables as a result of some process. This is very scalable since it's not precise, like which the vegetables are, for instance. If it's in that category, this retention factor applies. So it's scalable, but it's not precise. You end up invariably over or underestimating something. So the solution hypothesis is that there is a learnable pattern in composition data. So what happens before and after? In this case, it would be raw and cooked, but there's a pattern in all of the components in the before-after state when you've got enough number of these samples of raw and cooked, that there is a pattern and it can be learned. And this has been proven before in some work. There's been several different projects that predicted missing nutrient concentrations in a composition data set. These predicted 1 or 2 nutrients, so it's not at all what I propose to do, but it does go to show that there is a learnable pattern. And then there was another work that predicted the degree of food processing. from composition data. So didn't predict anything about the nutrients, but given some nutrient profile, it was able to predict one of these 4 categories. There have been also nutrient prediction models. Again, they were slightly different. So which kind of goes to show that prediction is important. It is something that's being looked at. So that one predicted it from the description of a food, so it was natural language processing, and in another one it was predicted from the ingredient list and limited number of nutrients. This prediction model is going to predict X is the input to the model and Y is the prediction from the model. So you're basically saying Y is a function of X. And the X is, there's 27 components were selected. So there's many more components, of course, in the food. But here I selected 27 components. And these were various vitamins, minerals, cholesterol, water, fat, carbohydrates, and proteins. And prediction is of these 27, 7 vitamins and 7 minerals in the cooked food. There were 425 pairs of raw and cooked food. These were plant-based as well as animal-based, and I'll get into what those foods were a little bit later, and 5 processes. So selected simple processes and just a simple process on one food. So, nothing like pizza, nothing with multiple ingredients or multiple processes. This is an overview. So you go from left to right, and I use the USDA data set, and I use SR Legacy, and from SR Legacy filtered out, curated these raw and cooked pairs, and then took the nutrition information, selected only 27 of the total nutrients, because they were they had data for 80% of the foods. And so I didn't want to drop below that threshold. And now an important thing is that there were no processing details. And so I divided the processes into and the data into wet heat and dry heat, and separately trained 2 models. I also tried training one single model, but the accuracy wasn't very good. So then the machine learning pipeline was used to fit this data, and out came 27 different models. So it was each micronutrient had its own prediction model. Again, it wasn't possible to predict all of them, and I can get into that later. So there were one model for each nutrient, and also they were divided by the wet heat and the dry heat processes. And the evaluation metric for precision was the root mean squared error of the predicted nutrient content compared to the true data that was in SR Legacy. So this is the composition data from SR. 178 plant-based pairs and 247 animal-based pairs. You could see the subcategory division there on the right, and it was unequal, which is just information. I mean, there's no comment on that. And here are the 27 components for the raw and the cooked food. So there is some breakdown across the fats, but for the proteins and the carbohydrates, it was just total protein and total carbs. Then the data representation, this turned out to be an issue, actually. And the reason is that the composition of a food, whether it's raw or cooked, is represented per 100-gram sample. And there, in this data set, there weren't yield factors. Yield factors are available elsewhere, but I didn't want to use, for this project, I didn't want to start combining data from different sources. So the problem here is that in dry heat cooking, food loses water. So the yield is less than 100 grams if you assume that the raw food was 100 grams. And so you see a concentration effect So you're scaling it up, so you're scaling it back to now 100 grams, and you see a concentration effect here because you've lost water. And when food gains water in the wet heat cooking, the yield increases, and that's because water enters, and you end up with a dilution effect. So, of course, you might lose some nutrients in this process, but also there's a dilution effect. So you end up with more water and less nutrients. So this leads to a data anomaly situation where the nutrient content in the cooked food is greater than it was in the raw food. Now, this happens due to the concentration bias in the dry heating process, because the nutrients get concentrated. So you could tend to see, when you're just comparing the before and the after, that the nutrients have increased in the cooked food, despite the fact that there should be less because nutrients were lost. I'm taking the simple assumption. You could argue, what if something happens and nutrients are increased, but I'm not going there for now. And the solution for this is that is a data scaling to reverse the effects, or to somehow undo or mitigate the effects of this concentration bias. The other reason is that the raw and the cooked samples were unmatched. So if you were cooking a potato, it could be that the raw and the cooked potato were not from the same physical sample, and there are inherent differences between the potatoes themselves. And there is no way to really undo this, except I'm just filtering. I'm checking if scaling can change anything. And if scaling wasn't able to fix the problem, I just take the pair out for that prediction. So these are data scaling equations. You don't have to try to interpret these equations. I could tell you what the intended effect is. The first one is basically, since we've, to address this dilution or concentration bias, the water entering or the water leaving, to equalize the water, to say the water is the same before and after, and then to basically then scale the rest of them proportionately. The second one, and you see this was specific to cholesterol, but the idea here is that let's assume that there was one ingredient, one nutrient that does not change. There's a process invariant nutrient, and it's the same in the food before and after. And so let's use this to get the scaling factor. So here we're actually trying to address the missing yield factor issue. So this is what happens applying the scaling. So actually the plant-based foods, that's the first figure on top. These were also all the wet heat foods. And I mean, which kind of makes sense. I mean, not that you can't roast it, but a lot of wet heat foods are a lot of steamed or boiled foods certainly include a lot of legumes and grains and, vegetables. So, and you see there's like 3 vertical bars there. The first one is unscaled, so the original data as it is. And then the scaling, keeping the water content the same. And the third one was the process invariant, where you're making this assumption that some nutrient remained the same before and after. And the height of the bar is that these were the number of foods where that didn't have this anomaly, where the nutrient content was less after. So that's what we want. That's good data for training the system. And you could see that there's really, I mean, when you statistically analyze this, there's no significant change due to the scaling. And for the animal-based foods, which were all dry heat process, where there was this concentration bias, and so here I would expect that the scaling would actually have a big difference, and it did. And so I forgot to add the significance, but this one was also statistically significant, and visually you can see that as well. The interesting thing was that the process invariant nutrient turned out to be cholesterol. that was the best, that also performed the best. So I scaled by all nutrients, and then I trained all the models, and actually cholesterol was the best. And from a domain science perspective, this is actually true. Cholesterol is not lost in the cooking process. And this is because physiologically, the way it is and my paper has a reference to another paper that researched what happens to cholesterol. So actually, cholesterol does not change. And by making this assumption, you could see the number of non-anomalous, the food pairs really increases, and also the performance is much better. So this was very interesting. And here are the results. This is a comparison or just showing the predictive performance. So just kind of adding, more information to the scaling effects. And you could see there at the bottom that the prediction, the error is the least for the dry heat processes. The error is the least in that last column there, the process invariant nutrient scaling. There were a few other nutrients that also had quite a sort of a comparable or even less error. Was protein and iron. And protein's interesting because it was basically a measurement of the nitrogen. And iron's interesting because for this data set that I used, the SR data set, actually the experimental results showed that iron was also process invariant for those samples. So you don't want to boggle you, all this data is in the paper, but here's just looking at the errors for the predicted versus the baseline, and the baseline were the USDA retention factors. RF100 is just to say that nothing changes, input is the same as the output. And this was just like a very, very baseline sanity check that, and it's only that if that ended up being the best performing model, then, the prediction model was of no use. And thankfully that wasn't the case. So the prediction model is better than the baseline in 22 out of the 27 tasks. 27 tasks being the 14 models for the wet heat, and then the 13 for the dry heat. And overall, it was 31% better, but it was 43% better for the wet heat and 18% better for the dry heat. It's hard to explain why this was different. So again, this is just reporting the results. These are the scatter plots that show the four on the corner. They show the true value on the x-axis, and what then the predicted, the machine learning model or the baseline value on the y-axis. And here you see in the top two figures, the prediction is better than the baseline. And I wish I had an x equal to y line actually here, because then you could see if there was an x equal to y, which is the ideal case, which is what you want. You want the true value, or the predicted values, to be as close as possible to the true value. The prediction actually falls on that line, close to that line, whereas the red dots that are the baseline, they go almost up to 2 times the true value. And the lower two plots is where the baseline was actually better than the predicted, but you see it was actually by a very small margin, because there's kind of a near overlap of those gray and red dots. Then, having seen how prediction was better, I thought to go into each of the different food, plant and animal-based foods, and see if, again, you could see that prediction was there was a difference between plant and animal. And then I thought whether, but if you go further subcategories, if there's a difference again in where the prediction performance was better. And so I divided all of the plant-based foods into these categories: green vegetables, root vegetables, just all other vegetables, legumes, and cereals. And the prediction was the best for leafy greens and the worst for cereals. And the second one, and sorry for the missing headline there, but for the plant-based foods, the prediction performance was the best in beef and the worst in veal. And again, this is just an observation, but for the plant-based foods, it would be interesting. What this basically means is that if we want to do experimental data and collect good composition data that we can then train models on, it would make more sense to focus on where the prediction wasn't good, because what it could mean is that there's so much entropy there, there's so much variation there that the predictability is low. Whereas for green vegetables, that there isn't much entropy or there isn't much variation. And because of that, that means everything is like everything else pretty much. And so the prediction performance is very good. So finally then is to see whether we can say something about the features, which feature is important to the prediction result. So here you see two examples: vitamin B6 and potassium. And the features there are all those 27 different components or compounds that were the X. And so it's interesting. Again, this was just an observation. It was hard to draw any domain conclusions from this. But vitamin B6 had only 3 features. And so as you add more features, the performance drops off. And for potassium, again, it wasn't a smooth curve, but there was 14. So these results were very specific. It was hard to do any domain discovery from this, and I'll get to that later on. And these are the actual features. So the one thing that was consistent is the highest ranked feature was always the self feature, which makes sense, which is to say that what happens to some nutrient in the cooked food, the most important feature is that same nutrient in the raw food. And again, this could be, if you've read the paper, then you would know, but otherwise this could be too much information in one plot. But this was basically taking an average rank across all of the different features. So there are 14 predictions, and in each prediction, a certain feature could have ranked higher. And so if you take the average rank across all the predictions, you see that generally the average rank, so you see the box plot there, but then you also see the averages, and they are in the mid-range, which means nothing was very important or very unimportant. And there's no information here, but this is interesting, like nothing really stands out. So if one needs more information, then looking at everything is probably not going to give any information, and that would also make sense, because the way loss happens in different categories, as you saw earlier, the way the loss happens in green vegetables or in grains or legumes is different. And so it actually makes sense. This is consistent with the previous result, that there is no high ranking feature across all the predictions and all the foods. So this is a review of the results, that even though there are many questions that are unanswered, but the results are interesting, and prediction does offer some advantages. It is multi-parametric. Again, this was limited to the dimensions where we had data, but the fact is that it is multi-parametric. It is precise. It gets fairly close to what the true values are, and it is scalable. You can just keep adding more samples to this. But performance analysis still needs more work. Feature weights could be very useful for domain discovery to understand loss mechanism, or which feature is more important. And this could be also then useful to design nutrient loss prevention strategies, or plant breeding as well, if we find that we've indeed, discovered something about the loss mechanism. And nutrient loss patterns are also affected by food structures. And this was simply the content of every nutrient. It had nothing to do with whether carbohydrates are a kind of hindrance within the food matrix and somehow prevent the loss of a nutrient or something. This was impossible to tell. And this was just an example, but food structure could really have an effect. And so the unknown is, how do you even represent this? And then, of course, test it. And then there's a few more that get to the next slides. So are there any questions at this stage? Can I just keep going on? Okay, I'll just go on. So the baseline method, of course, is very, very simple, and it's not comparable to the machine learning method. It would have been interesting to have had a state-of-the-art machine learning method and then compare and say why this is better. But this was the first one of its kind. So it is a machine learning model, is sophisticated, is more complex, and the baseline is simple. And one of the limitations still of the baseline method is that there were no reference details for these retention factors. What were the vegetables that were used, or the cooking method? If it said, so much loss for this category for boiling, what boiling is, of course, 100 degrees Celsius, but what was the length of time? And so it could be that the retention factors were based on a different cooking protocol and the sample data was in a different protocol. And if that was the case, then it would not be a fair comparison, but there was no way to even know this. So there were, besides this missing data, there was also other missing data, where not every data point, not every nutrient content was measured experimentally. There were some that were calculated, but there were no references for how the calculation was done. There was no information to confirm whether the pairs were related. And of course, there was no process information. How were things cut or prepared? And there was only one of each plant-based food, so there was no food diversity information there. So these are some of the questions that I'm working on now in the next paper, and I'm happy to take any further ideas that you may have about this. So the first thing is to make these models scalable, and this means two things. What is the data size? I mean, there's certainly, besides the foods I use, there are also other foods that were not there, but even for, a potato or a carrot or something, how many more, how varied? And the answer would depend on how different are carrots from each other. So how many carrots do you need? And for features, is this scalable? For the features, can you predict polyphenol content similarly using this model? Then is this reliable? And what that means is, can we reduce experiments and just use machine learning, or can we at least use it reliably for simulation? And do we need experimental verification to first verify that these precision results, these performance results are correct? And then we could decide whether, we can use now, because of course it's much cheaper. It takes away so many of the hassles of experiments. And then the third thing is, how can we represent food structure in such a model? So food structural detail is not there, but is there a way by which we can still represent it? So how do you set up basically an experiment? And one idea I had was to compare cooking from raw to boiled or steamed versus starting from frozen and then cooking it, and then seeing if I could just simply apply the same model or retrain a model when the food was frozen. And then it would be interesting to see if I could compare the features or the feature weights or something and see if it was different. Then lastly is integrating data sets. So if you want to put together the same data set that basically has the same sort of information across different sources, can you combine it? Is there a quality check that you would have to do to be sure that you don't result in a garbage in, garbage out phenomenon in your prediction models? And then the second one is a much more complicated question. Could you basically combine everything? Could you combine data from mass spec, other spectral methods, NMR? Could you combine all of this data? And along with this composition, this nutrient content data, is there some way in which you could put everything together? What sense would that make? And then finally, which is not here, but I'm working on this, is how do you mine a data set? So when there are lots of data sets available, how do you select the data that's going to be relevant for your model? How do you avoid garbage in, garbage out? Yeah, so someone have a question? Oh, sorry. And I think that's it, I think.
Okay, so wonderful stuff, Tarini. Already got a few comments, one from Jaspreet in the chat board, and then a couple of hands up. So shall we just take a look? Jaspreet, you're online right now. Would you like to speak to what you wrote, and then we'll move to the questions?
Sure. It's more of a comment, I would say. And I understand, the need for established data sets to be used for training, and, probably would have done the same. But I just wanted to point out, as a defense of the SR and the USD databases, which I worked years on, that they do say that, the data from different forms and products are not from the same sample.
Right.
So they're not from the same sample or source. So, for example, they just give it this example of wheat. So you can't really say that, because there's so many variations that go into. There are so many potential variability factors which can go into determining the nutrient content. Like the cultivar, the location, the crop years, and since it's not the same sample, so to say that the processing is the cause of the difference between the two is not what the SR suggests that we do. So it's not really the effect of processing. And another thing I was going to ask is, did you get a chance to look at the FNDDS, which is a database which is also part of the FTC, where there is yield factors? Because I think one of the things that you were saying was no yield factors obtained, was available. So which does have yield factors and retention factors applied to determine. So they'd use the calculation method, where it can take raw food, apply the yield factors and the retention factors to determine the nutrient determinations for a cooked product.
Yeah, actually, that is a next database that I want to look at. I was talking to Alana about FNDDS, and I did want to look at it. And yes, to your point, of course, things can vary. All these growing conditions have an effect. And I wanted to I'm still curious to see what is the variability. Does everything change? Is a carrot nothing like another carrot elsewhere, or is the change specific to certain molecules? And this is not to say that, not to defend my model, but more just to see that what can we learn from the existing data that is there? How do we understand and how do we prevent garbage in and garbage out syndrome? Because it could be that there'll be, someone else that just does pure machine learning and they don't understand the complexity of food. And you could end up with models that are, promising great results, but they don't inherently, they miss something about food complexity.
I totally agree on that. Yeah. And another a possibility, which I'm not a computational expert or anything, but looking into taking that variability information that is available on SR Legacy and more so even now in foundational foods. And I see Carl's there, so he can add to that, is that we have more variability information now, maybe taking that as aspect into the machine learning process, not only just taking the means, but taking that the variability information also into the machine learning models, I think would be helpful too.
Yeah, that's actually the thing I'm doing in my next work. And I'd spoken to Kyle about discussing this because I do want to make, as many considerations, and because this is your data and you know a lot about this and the conditions under which you measured this data or calculated this data. And so I would like much more awareness about, these complexities in designing the next model.
I might tag on here. Tarini did consult with myself and Pamela at the USDA in regards to these data sets. And Tarini, I think, has a very deep understanding that there is high quality data in SR and it's a great data set, but it does have limitations. And we're aware of those too in regards to documentation or really understanding around these. But one thing I want to note on these that is to maybe also say what Jaspreet was saying here is these SR samples were composites. And so they are sort of better at representing the average value of a particular food. Same with the cooked versions as far as better representing an average. And so I think it's really interesting to see how well the model did on the vegetables than it did on other products, given that they are supposed to be average, that wasn't the case for some. And I am really interested to see how that model potentially expands with the new approach of foundation foods being and focusing more on the variability within a food and not necessarily that average amount. And then how much does that actually matter, or what does that look like, or how does the model look like when you add that actual variability from the raw version, even though you don't necessarily have cooked single samples at this point.
Yeah, I mean, I also think just, working with new data, running the models, kind of doing this in a cyclical pattern, you might be able to find areas where you might want to focus on. So if you had a question about, should we measure this or this, then, if you want to look to predictive modeling as a metric for, what you should measure, then you could use this in a cyclical way like that.
Just before we get to Matthew's question, my question is the foundation foods. Are you doing possibly testing of boiled and raw samples? In other words, finally getting to this golden day where we actually know what the input sample was in both cases.
Yeah, and Jaspreet and Pamela's group are going to be dealing with the changes in cooked nutrients and whatnot. What we have said is that when we do cook future foods to have that analysis in FDC, is that it will be within the context of experimental foods and not foundation foods because of some of those items that Tarini mentioned when it comes to temperature and length and whatnot that are more difficult to discern from SR is making them much more clear. In fact, forcing you to recognize these as we're in a research kitchen, not your oven at home, not your stovetop, and some considerations as far as how you're using these values. And so they'll be in experimental foods, not foundation, but sort of still map the same. As far as what's going to be cooked and whatnot, that's more Pamela's group within MAFCL to sort of make those decisions.
Okay. So Matthew, what would you like to ask or comment on?
First, excuse me. First, I just want to say, great job on really. I think you've laid a foundation for a way forward, a new methodology that is going to end up saving a whole lot of money for a whole lot of people. So that's awesome. And I'm not sure that my comments are going to be relevant. I did read your paper, but just tell me if I go astray. But it seems like there might be a way to build on top of this with information that already exists in a couple different ways that I see. So one is you pointed out that leafy greens prediction performed better than legumes, for instance. And I wonder whether that might be because of the nutrients that are often associated with leafy greens, in that B6, B12, the various B vitamins, they're all relatively heat labile, so they would all degrade at relatively the same rates, whereas what you're looking at inside of the pulses or legumes may be a more of an array of different kinds of nutrients. I'm not sure if that's what you were saying already, but I think that.
No, I used the same features for everything, right. All the predictions and on the outputs. So, I mean, what you're saying is like domain knowledge. This is what we know from all the experiments we've done. So you could certainly like use that, but the problem is that the feature weights are not really reliable because of, so many problems with the data sets. So you could confirm domain knowledge with this, but no, I mean, the data set wasn't biased in any way on domain knowledge, and which is what makes this, which is what you would do when you design a machine learning model, is you don't bias it. You don't say, this is what I know, and this is what I'm going to test for. You just test for everything. The way I tested also for process invariant nutrients, you just basically test for everything. You find the best results from machine learning, and then you see, does this confirm what we know about domain, or does this provide domain discovery?
So can you then go back with domain knowledge and add weights to your features to improve prediction?
But we don't really, I mean, you could start with a pre-trained model, certainly, but you would need to really know what those weights are. You would need to know when those weights are relevant for which categories, not relevant for other categories. So pre-trained models exist, but I think what you're saying is a very simple way. Like if I wanted to model only leafy green vegetables and that was the only domain knowledge I have, I would need to know for a certain feature X. If I wanted domain knowledge, I would need to have that domain knowledge across all of the samples in my training data. And right. I cannot skew it like that. So it's almost better to start at this stage where we don't have that kind of consistency in domain knowledge. It would not make sense to initialize any of the weights.
Is there
just one second, Damien. Let me just follow on with that. So what I was thinking was, although the example I gave was the leafy greens versus the legumes, what I was wondering was, and sort of the second part of my question was whether you could take the domain knowledge about the nutrients themselves, not the foods, but the nutrients. So, for instance, adding information, say from PubChem, about the nutrients, about the temperature at which specific nutrients degrade, and whether then seeing, well, which of these foods are enriched in these nutrients and therefore are more likely to change.
Well, you would need processing temperature for that. And basically oxidation happens anyway, even at a slightly elevated temperature. So yes, you could, but you would need a lot of information if you want to start adding information about only a few pathways. So I think that the level of detail that you would need to start adding domain knowledge is a lot.
Okay.
I was just remembering one approach to looking at data sets like this, where you might run the little machine learning models on each particular nutrient with respect to a food, but then you start looking for models that start to group the predictive success. So starts to identify the things that are performing really well versus those that aren't, into little islands. I forget what that's called though in the machine learning business. It's something about bootstrapping. But anyway, it was just a straight thought. Do you know?
No, I tried bootstrapping. It didn't improve the results.
Okay. One other question is whether spectronomy, mass spectronomy might help provide some experimental results. Is that a much cheaper technology to get nutrient information out of foods in their uncooked and cooked state, such that, like, I could cook something in my kitchen and then hand in both the raw carrot and the cooked carrot to some lab that did spectronomy and got a result right away? Is that like an order of magnitude cheaper than doing the other kind of laboratory?
I mean, I think some of the more the experts in experiments could probably answer to this. I know there's a data set, PTFI, the periodic food table, they're using mass spec, but I believe, and please correct me, if you know more, it only gives you the nutrients, like you can identify the nutrients, not the contents, or the molecule, but not the content.
Okay.
Yeah, I think it's going to, if I might there, yes, PTFI is going to be that. We are working on having a summarized version of that on FDC with sort of a link back to the raw data, if that's what you're into. But to what Tarini said, that's going to be semi-quantifiable data. So that's really going to be sort of better values within the reference of the rest of the PTFI data set, because you're using the same methodology consistently. So it's going to be better to maybe compare that data within that data set than it is to sort of directly link it over to the quantifiable data set like Foundation Foods. But it will be an interesting data set. Hopefully in the next few years, we start seeing some more of that.
Right. I guess it has to prove itself. Will be to do the comparison to Foundation Foods will reveal for the very same thing, whether it's coming up with different results.
Right. And they'll be measuring some different things, but we are going to try and have some samples of our Foundation Food saved that we know the results on our end to be sent through PTFI as well. So we have some interesting things coming up.
Right. Okay, Troy, you're rumbling.
Hi, guys. Enjoying the discussion. As Damien alluded to, and I know, Tarini, you had to grapple with, it would be so nice if we had these golden pairs where we knew it was exactly the carrot from that lot raw, and then the same carrot, or the same kind of carrots cooked. And so we would like to be linking the samples, in this case, raw and cooked. And of course, you could imagine lots of other cases where researchers want to link things. We want to know whether the same thing has what the variability is of some other characteristic for the same food, for example, or for other multiple foods that come from the same farm, or what have you. And so I'm just thinking a little bit about matchability. And so if we have samples and we have ways of tagging these samples, ideally we'd like to be able to draw connections between these samples from their source, from their other attributes. So Tarini, to you, and then maybe to Damon and others, I'm wondering, what have we learned through this about the taggability or the identifiability we would like? Yeah. Of course, if each sample had a number and you could find two samples with the exact same number, great. But that's not usually going to be the case. So normally you would say, well, two samples that came from the same geography or were gathered at the same time or what have you, that are proxies for sameness. So I just wanted to put that question, maybe Tarini, to you first, and then maybe to the broader group about what we've learned on that dimension here. That's not about nutrition per se, right? Or about cooking per se. It's about identification of individual samples.
Well, I mean, if there was no documentation on this was the same physical food, then it's hard to say why they were not the same. I mean, were they like, grown in two different geographies? Like, it's really hard. I mean, if you have like one sample of carrot in your data set, now if you decided to, let's say, attribute variation to, some one of, let's say, 10 factors or something, you'd need to collect a lot of carrots, train it. There's no magical easy way to just figure out what's the difference between these two. It all depends on how much training data you have and the patterns that you've learned. So yes, I mean, in future, if you wanted to develop a set of sort of QC quality check protocol for data, is this data useful for this or not? I mean, you're basically talking about a massive data platform with toolkits that have been trained specifically. So if you could think about this as a massive Python library or something, you would need a carrot module where input 2 carrots. Can I use this? And what are you doing with it? Are you doing nutrient analysis? Are you doing texture prediction? What are you doing with it? So I have these two. This is what I'm using the data for. A QC question, can I use it or not? And so, of course, I mean, the day that we've got tons of data, we probably have a massive Python library where we can ask all these questions because it would have been trained to answer these questions. So if you don't, the short answer is, if you haven't trained the answer to this on data you have, you're not going to get it.
Yes, I guess what I'm thinking is, realistically, the answer here is probably metadata. Yeah, in other words, the more metadata we have about our samples, the more variables we can potentially control for. I guess maybe that's the other way of asking my question is, what is this teaching us about the minimum necessary metadata for conducting reasonable comparisons between things? It's too abstract of a question to answer in general, but yeah.
I mean, Carl can say something, but I think minimum necessary, it's very complicated, right? At some point we need to have reductionist approach because we cannot measure everything about everything. And slowly we start this reductionist, this limited principle approach. We start relaxing these principles. So I think that we can't immediately attack this question about let's do everything on everything. We've got to start with certain experimental limitations because this technology is still expensive. So it has to go, I think, step by step.
Mm-hmm. I'll just say a project that Graham Cross, Liliana Hernandez, and Marie-Angelique Laporte, and I, and others who are interested in, is actually coming up with the biosample from the field all the way through the chain, coming up with a minimum set of requirements for information around that. Of course, the minimum depends on who's looking at it, but at least trying to draw upon the various ontologies like agro for the dimensions, such as organic farming practice and other kinds of field practices applied. So that's just to say, without any standards of data collection, it'll always be looking back to see what you might salvage from any given data set.
Yep. Maybe while I've got the microphone, maybe one other just quick comment. Tarini, I am really excited by the idea. I think this is what you were positing, that we could add to the body of knowledge on retention factors using the calculated results from a model like this. Yeah, that you've calculated a version of retention factors. There's all kinds of reasons why sometimes those numbers are going to be too high and sometimes they're going to be too low, but there is a source of truth out there now, a reference table. You've created another way of doing that. And I would love to find a world where we can add that up and say, look, here's just another way of another version of truth on this, and let the body of knowledge, in this case on retention factors, grow and grow. So then people can do meta-analysis on those and what are the drivers that seem to be, which ones seem to be better and when. So it's exciting. It's cool stuff.
Yeah, it's good to find out this process invariant nutrient, because we know it's a domain discovery thing. It tells us something about the physiology of, plant cells, or, whatever, the anatomy. And it's a really great way of using old data where we hadn't recorded this. So I think so it's not just a criticism, but it's good to collect this information and reuse data that otherwise wouldn't be. But I mean, there's so much more you can do. Like, this is my next project is texture prediction of carbohydrates, predicting dietary fiber content after you cook and store something. So yeah, I think, standardizing food composition data can be used for many things.
Right. Any other comments on this before we wrap the hour?
Oh, just does anyone else, or can I just briefly say? Yeah, so that's one of the things in terms of metadata and trying to process all this stuff. That's one of the goals that we have now. So I've just started with USDA. I'm a postdoc there now, and we're trying to have a better representation of a lot of the different types of data that might be in SR or the various other databases. And so it's a step toward making things like this possible. So the goal, in long and short, the goal is we want to create systems on top of the existing data where we have better faceting of the existing data that makes it searchable, such that you could ask a question like, what X data fit these categories, and just automatically deliver that data. So systems like that in the future could be used to help facilitate machine learning models like the things you're trying to do here. But there's that's a whole nother set of work to get all of that stuff cleaned up and machine searchable. So that's kind of another analogous way. That's I think a orthogonal path to getting to helping answer those types of questions. But there's a lot of back-end cleaning that has to happen to make that data available and useful for machine learning models in the future. So yeah, just my 2 cents there.
Yeah. And in food on too, for example, we'd love to have the linkage of food process boiling between the raw and the boiled spinach and so on in an actual relation within the ontology, like you had to manually compile to get your work done. Okay, I'll just wrap that. We don't have anything lined up for December. We wouldn't want to make it the last week in December for a joint call. So what I suggest is, if anybody has an idea for presentation, I'll put this out in an email for before Christmas, that last week before Christmas, then we might have a joint call then. Otherwise, let's collect ideas for presentations for next year and create a schedule, get a schedule going for that. Okay.
Okay.
Great.
Thank you
thanks
for your
so
comments and feedback, everyone.
Thank you. Great job.
Yeah. Wonderful. Okay. Bye, folks.